Hello everyone, we are from Structural Biology BSN 4603 Group 1 SCL. In this video, we are going to discuss protein-ligand interactions of leptospira chemically riboflavin synthase. So this is our team. Molecular docking is a computational technique that predicts the binding pose and affinities of a small molecule, which is ligand, to a protein active site by using in silico simulations in order to providing valuable insights for drug discovery and rational design of new therapeutic, although its accuracies can be limited by complexity of biological system. Molecular docking predicts how small molecules interact with proteins binding pockets. Protein-protein docking is typically much more complex because proteins are flexible and their conformational space is quite vast. Docking can be executed by inserting the inflexible molecule or fragments into the active site of protein through various methods. The effectiveness of docking relies on the performance of the search algorithms. In this study, we are going to use our target protein, which is riboflavin synthase from Leptospira chemically. This protein is important for biosynthesis of riboflavin, also known as vitamin B2, which is essential element for Leptospira chemically survival. Leptospira chemically is a gram-negative bacteria which cause leptospirosis disease. So in this study, we are going to use 7 potential inhibitors and 1 control inhibitor, which is the potent inhibitor in order to understand how they interact with riboflavin synthase at the binding pocket. The problem statement revolves around the need to enhance the efficiency and accuracy of molecular docking algorithm and also is crucial for predicting optimal ligand conformation within a protein's active site and thus addressing challenges in computational biology and drug discovery. The objective of this study is first to retrieve ligands from online database and second to convert the format of the ligand file and provide hands-on approach to molecular docking and last but not least to analyze the receptor-ligand interactions.
Now I'm going to present about literature review. The global threat of antibiotic resistance demands urgent action. For powerful bacteria emerge and outsmart existing drugs. Scientists find hope in targeting a unique vulnerability, the riboflavin biosynthesis pathway. This pathway, absent in human but vital for certain bacteria, it presents a promising avenue for new antibiotics. This study focuses on Leptospira cameri, a bacteria responsible for the neglected tropical disease leptospirosis in Malaysia. By revealing the structure of its riboflavin synthase enzyme, the researchers paved the way for designing drugs that specifically cripple this vital bacterial process. So this targeted approach holds immense potential in combating rising antibiotic resistance and protecting against diseases like leptospirosis. So next, my friend is going to present about the methodology.
For the methodology, the first step is to access the PubChem database, searching the potential ligands that given and download it in a 3D structure and save as SDS file. Then, use the OpenBabel to convert the ligand file format from SDS to MOL format and save it. After that, we need to generate the binding site without the ligand. So open the Pymo apps and load the crystal structure of LKRS. Prepare the binding gravity for LKRS by selecting the residues around 12A and save it as cell PDB. Next, run the iGEM for docking simulation. After the docking is complete, display the docking energy and types of interactions. After that, use the Pymo and route the binding gravity with the top ligand and repeat the same step for LKRS ligand. Finally, use the LibPlot software to generate the protein-ligand interaction.
Okay, so next, let us proceed with our results, with our findings. So, after complete docking by using iGamDock, we found that zinc 594-20747 show the highest binding energy, and this show that the zinc 594-20747 has the higher affinity towards the binding site. And for the reference, we are using the potential inhibitor, which is provided by doctor with the energy of 105.67, and the one we're saying that with the higher binding energy is the zinc 594 with the energy of 10120.15. But we also found that there's another potential inhibitor which have the higher binding energy compared to the potent and inhibitor provided, which is the second one, which is the zinc 128.94491 with the energy of 108.63, which is higher than the energy of 105.67 given in the potent inhibitor. And in these slides, we are looking for the interaction analysis for the docking results. And here we can see the green color and also the gray color, right? So for the green color, it is actually referring to the hydrogen bonding. And also the gray color is referring to the van der Waals forces. And also we can see all the energy in here provided in every potential inhibitors. And we can discover that there are some tick at the top there, right? And the tick of at the top there is actually the consensus sequence. And the consensus sequence is actually which is the most frequently occurring residue at each position within the multiple sequence alignment.
Okay, next, let's move on discussions. After the docking process, we found that ligand zinc 59420747 has the strongest overall predicted binding affinity due to his combinations of strong hydrogens and van der Waals interactions. Thus, it binds strongly to binding cavity compared to other ligands. Based on all ligands protein interactions individually, we found that the consensus binding residues of the protein are serine 141 from the chain A and glycine 97 from chain A. We have two ligands which bind better than positive control. The first one is our top ranked ligand, zinc 59420747, which has been mentioned just now, and the other one is zinc 12894491 after the docking process. So these two ligands show higher binding energy and van der Waals energy than positive control. Therefore, we have said that they have better binding affinities than positive control. These are the two images which show the interactions of ligand zinc 59420747, which is our top ligand, with the protein. SELE ligand interactions refer to interactions between the ligand and chosen residue. In this study, the residues are selected around 12 angstrom from catalytic residues, histidine 98, serine 148, and aspartic acid 187 of chain A. LKRS ligand interactions refer to interactions between the ligand and the entire protein. There is a slightly differences between these two images in terms of interactions. LKRS ligand show 5 hydrogen bonds and 12 hydrophobic interactions compared to SELE ligand, which show 4 hydrogen bonds and 11 hydrophobic interactions. The possible reason which lead to this outcome could be selected residue might represent a non-binding pocket, but the ligand could be interacting with additional, less characterized pocket on the protein surface. These additional interactions would not be included in the selected residue analysis, but could contribute to the overall stronger binding energy observed with the whole protein. Despite the slightly differences, overall, zinc 59420747 show a good interactions with riboflavin synthase.
Differences in ligand binding affinity toward binding sites of the following riboflavin synthase protein that is shown is contributed by hydrogen bond, which is the electrostatic interactions between partially positive charged atom, hydrogen, and partially negatively charged atom, nitrogen or oxygen, that contribute to overall protein ligand complex stability, while the high specificity and selectivity of binding sites toward ligands might be the result of the narrow range between partially positive charged atom, hydrogen, and partially negatively charged atom, nitrogen or oxygen, which is 2 to 4 angstrom, as well as well as the strict requirements of nitrogen and oxygen atoms for the formation of hydrogen bond. Hydrophobic interactions and other driving forces that facilitate the ligand binding involves the exclusion of free-moving water molecules by the binding sites and the hydrophobic surface of the ligands, which will increase system disorder, then followed by the increase in the entropy, and hence and favor the ligand binding event. Whereas the binding site specificity toward ligands is the result of the requirement for the size and shape of the hydrophobic regions on both the ligand and the binding site to be complement. Sometimes hydrogen bond and hydrophobic interactions can work together to further stabilize the protein ligand complex. Here are some identified potential amino acids residues which might involve in the protein ligand interaction. For hydrogen bond, serine with side chain hydroxyl group, aspartate with side chain carboxyl group, and histidine with side chain amino group. The carbonyl and amino group of all the backbones amino acids could also have contribute to the hydrogen bonding between ligand and the protein binding site. While hydrophobic amino acid such as leucine, methionine, isoleucine, valine, and tyrosine might have involved in the hydrophobic interaction formation in the protein ligand complex. Ligands that mimic the structure and charge distribution of natural substrates, which are PRPP and air, attach to the protein's respective binding sites, blocking the protein substrates interaction. This prevents the catalytic process from occurring and thereby interrupt riboflavin production. Slight conformational changes caused by ligand interaction may also limit enzyme activity, revealing the protein flexibility. The RPP binding pocket, which is found located at the N-terminal domain of the protein, is responsible to house the PRPP 5-phosphor-alpha D-ribosyl 1-pyrophosphate molecule, a vital substrate for riboflavin biosynthesis. These binding sites are hindered from PRPP by the binding of the ligand, thus interrupt the enzymatic riboflavin biosynthetic pathway. Air binding pocket is the binding site located at the C-terminal domain of the protein that accommodate the air, which is 5-amino-imidazole ribotide, another essential component for riboflavin biosynthesis via the enzyme's catalytic mechanism. The whole riboflavin enzymatic synthetic pathway can be halted by blocking the air from binding to this binding site via ligand binding. The most important catalytic cleft, which holds both the PRPP and air for the condensation process that will then lead to the biosynthesis of riboflavin, is found at the interface between the N and C terminal domains of the protein. Therefore, the bacteria's ability to produce riboflavin and grow can be terminated by focusing on the disruption of the enzymatic activity that occur in this binding site via ligand binding.
In conclusion, our in silico docking study identified zinc 594207747 as the top ligand with promising the binding affinity and pose within the protein's active site. Protein flexibility, solvation effect, and the potential interaction with other biomolecules can all influence ligand binding in vivo. Therefore, further study using complementary techniques such as biochemical assay and structural biology approaches are warranted to fully validate the potential of zinc 594207747 and gain deeper insight into its binding mechanism.